Hello everyone, welcome to ITLS Academy. This is your mentor Dr. Shamaita Basu, and in this session we will be starting with the next module or the next session of this course of forensic microbiology. So as we can see over here, so in this session we will be covering the topic, or we will be starting with the topic of history, current and future use of microorganisms as physical evidence. Physical evidence as physical evidence of forensic sciences, or of criminal scenarios, or like crime spots. So in this session we will be covering, I think, again, this is a topic which will need at least 2 or 3 sessions. So this is the first session of this topic. Okay, so let's move forward. So firstly we will start our discussion with why and how are microorganisms used in forensic science. Means what will be the role of microorganisms, or what like why are we even talking about microorganisms in forensic science. So, if we talk about molecular epidemiology, then it has been used for years to trace outbreaks of microbial diseases. However, what distinguishes forensic microbiology from molecular epidemiology is that forensic microbiology data must hold up not only to the scrutiny of scientists in the healthcare community but also to the scrutiny of judges and juries. The push for the increased utility of microorganisms in forensic science intensified as the fear of biological terrorism and biocrimes increased after the anthrax attacks in the United States in 2001. So first let us talk about what do we mean by molecular epidemiology. Okay, so I will just highlight it for you. Okay, so molecular, by this term molecular epidemiology, so basically we are talking about the the genetics part or the molecular basis of epidemiology. And what is epidemiology? Epidemiology is how and where a disease is spread. Okay, like what is the course, or what are the chances, what are the factors which will be governing the the spread of a disease, what are the areas the disease will be spreading, how the what will be the pattern of the spread of the disease, which which type of population or which level of population will be the most affected. So this is what do we mean by epidemiology. And when we go to the root level, to the grassroots level, for any disease, for any condition, for any like epidemiological reasoning, the ultimate root cause or the ultimate root level is the the genetics or the molecular makeup. So that is what we mean by molecular epidemiology. So now what is the basic difference between forensic microbiology, like why are we even talking about this term molecular epidemiology in this course of forensic microbiology? Because when we talk about molecular epidemiology, it is only important for the scientists in the healthcare community, because it's totally a kind of a research based thing. Because when we can like as scientists or as researchers, if we are the scientists or we are the researchers, then our main motto or main aim behind our research or aim behind our work is to dig deeper and find the the deep rooted causes, or the the basic causes, the main causes. So for that, whenever we are dealing with a new disease, say for example covid was entirely a new disease for us. So sorry, so whenever we are dealing with such diseases, if we are the scientists or we are the researchers, so we need to dig deep to find out the the basis, or to find out the main reason, because then only we will be able to come up with a cure, or come up with a vaccine maybe, or with a treatment, with a medicine. So that's where the role of this molecular epidemiology comes in. So that is where only the scientists community or the researchers are involved. But when we talk about this forensic microbiology, it has also it needs to come to the scrutiny of the judges and the juries also. Because whenever we are talking about forensic science, we are basically talking about crime. We are talking about the crime scenarios, the crime locations, we are talking about the victims, we are talking about identifying and catching the criminals. So we need to satisfy, or we need to come with that logic, uh, which will be acceptable by the judges and juries also. Okay, so and this, like, from where this concept of forensic microbiology, or from where this, uh, the necessity or the importance of studying these microorganisms in forensic science came from. So they have just given an example over here. I will again just highlight it for you. So it's basically, we can say it's a historical event, because like, it might sound that 2001 is not that much far behind, but it's actually very much far behind. 2001 means we are talking about 24 years ago, right? Because we are right now standing in 2025. Even 2025 is almost over, so it's like around 24 to 25 years ago we are talking about. So obviously we can say that it's a historical event. So what happened, we first came up with this, with this idea, or with this, we became aware about this concept of bioterrorism or biological war. So basically what happened in this anthrax attacks in the US in 2001, so some letters were posted in some US, in some specific US addresses, and those letters actually contained anthrax spores in the form of powder. Now, what is anthrax? Anthrax is basically a bacilli. The entire name of the bacteria is Bacillus anthracis. Okay. So, what this Bacillus anthracis do? It's a pathogenic bacteria which is responsible for causing anthrax, which is a particular disease. So, this entire event that which occurred, as I mentioned, that letters were delivered which contained this anthrax spores in the form of powder. So, that entire event is also known as Amerithrax. Okay, so because it occurred in the America. So as a result of this delivery of these letters, many people were infected with this anthrax bacilli or Bacillus anthracis, and they contracted this disease anthrax. So obviously it's a kind of bioterrorism, because someone had the intention to spread this disease in the United States, and that's why they had mailed those letters containing the anthrax spores, isn't it? So I think, like, what they're trying to say over here, that it was a major incident, or a major hallmark, you can say, which actually necessitated, or which actually instigated that we also keep in mind about the microorganisms, or we also give importance to the study of these microorganisms as far as this forensic science investigations is concerned. Okay, so let's move forward. Now, there are different methods by which we can actually identify these microorganisms. So, obviously, firstly, we will be talking about some classical methods. Okay, classical methods means we can also term them as traditional methods. Means age-old methods, जो पहले से it's going on. So I think one of the most classical methods or traditional methods will be just plain and simple culturing the bacteria or culturing the samples on nutrient media. And that nutrient media, as we can see over here, they have given diagram also. So it can be a broth culture, or it can be a agar culture. So this B image, as you can see, they have mentioned over here also. So this B is basically the the broth culture, or the liquid culture. By broth we mean liquid, so we can all see over here that it's a liquid culture. So the physical state of the culture is liquid form, right? We can do that also, or we can culture it in the form of solid media, which where we actually, the solidifying agent that we use is actually the agar. So basically, how we get this agar media or solid media, we just need to add this agar powder. It comes in the form of powder. It's also known as agar. It's nothing but seaweed. Okay, so we just add it in this liquid media. So whatever components or whatever constituents we have in the liquid media, it will remain the same. We just need to add this agar, and it's usually added in the percentage of 2. Like 2% agar if you add, then it will be enough to solidify that liquid media. Okay, so say for example, if I am like preparing 100 ml of liquid media, and if I add 2 gram of this agar powder, that is only 2%, so that will be enough to solidify that 100 ml of liquid media. Okay, so since it's the it's a solid media, so we need to pour it in these petri dishes that come. So this is basically how a typical culture petri dish or culture plate looks like. So we need to pour it and then we need to give some time, so it will automatically solidify because of the effect of that agar powder, and then we can also culture into solid media also. So the basic difference is that, like, what is the basic difference of these two media, liquid and solid, that also I would like to mention. So in liquid media, what happens after the growth of the bacteria, because it needs some time, it's not something instant that it will happen just like that, that I will just pour the sample in the liquid media and instantly they will grow. No, it takes some time, like usually it's between 12 to 16 hours. Overnight we have to keep the bacteria inside an incubator. So what the incubator do actually, it provides the environmental requirements that the bacteria needs. Say for example, the the humidity, the CO2 content, or the particular temperature. So all these are basically maintained inside the incubator. It's nothing but an instrument or machine. So we can accordingly set the temperature, the humidity, the CO2 content according to the need of the bacteria, because different bacteria has different requirements, different environmental prerequisites they need. So accordingly we can set the settings of the incubator, and then we need to put it inside the incubator, whether it's a liquid culture or the solid media, irrespective of that. So after that particular time, which as I mentioned, 12 to 16 hours, if we take it out, then we will see growth on this media. So if it's a solid media, as they have seen, they have shown over here on the left hand side A, so there it will appear in the form of colonies. So basically we can count, so say, as we can see over here, so we can count individually, isn't it, like 1 2 3 4 5 6. So these are basically the colonies of bacteria. So each dot means one colony, and one colony means there are many bacterial cells present, like maybe 1000 or 2000 bacterial cells can be present, or even more than that can be present in this single dot or single colony. Okay, but when we talk about the liquid media or the liquid culture, as we can see in this right hand side, here what happens, here we cannot count the colonies. So here, how will we understand that whether the bacteria has grown or not? Say for example, in the solid media, if we do not see these dots, if we do not see any of these colonies, then we can say that okay, no bacteria has grown. Means in my sample there was no bacteria. It's a negative sample, right? But in liquid media, how will we able to know? Because we cannot count the colonies because it's a liquid media. So here what happens, we can see the turbidity. Okay, so turbidity is basically the haziness or the viscosity. So when we actually inoculate the bacteria, means when we actually add the sample in the liquid media, at that time it will be a transparent fluid. Okay, so like the light will actually pass, we can see this side and that side. Okay, but after this 12 to 16 hours of time, when we take it outside from the incubator, then this turbidity will appear. So the image that they have shown over here, it is after the culture time, like after that 12 to 16 hours of time. If my sample is positive, means if some bacteria was present in my sample and I have inoculated the media, the liquid media, using that sample, then after this 12 to 16 hours of time, I can expect to see this type of turbidity. So see, this is no longer a transparent liquid, isn't it? Because some amount of dhundlapan, some amount of turbidity is there. So if we can see this turbidity, then we can say that okay, my sample is positive, the bacteria is present in my sample. And if we do not see any type of turbidity, then we will say that okay, my sample is negative, no bacteria is present. Okay, so the investigation is like nothing positive is found in the investigation. Maybe we will have to go in some other direction, like that. Okay, so let's see what they're saying over here. So the oldest form of microbial identification is by observing and biochemically testing a microorganism in pure culture. Various growth media have been developed and refined. These methods range from being able to provide nutrients to a broad spectrum of microorganisms to selective and differential media for the isolation of targeted genre or species. Growth media are usually found in either in an agar or a broth form, and the media chosen as well as additives for enrichment is dependent upon the desired microorganisms to be isolated. Okay, now the next method that we can go for is genomics and strain typing. Okay, so let's say what they're saying over here. So these are basically the different sequencing methods and applications that we can go for. So first we have 16 or 18S rRNA gene, that is ribosomal RNA gene, where the target will obviously be the ribosomal RNA gene, that is the rRNA gene. Application will be phylogenetic determination. Method summary: highly conserved rRNA genes containing slight mutations across species are sequenced for taxonomy. Second method that we can go for is SNP, which is single nucleotide polymorphism. Target will be point mutations. Application will be strain identification. Method summary will be single nucleotide mutations in conserved regions of DNA is sequenced for identification of strains within species. Third method that we can go for is internal transcribed spacers. Target will be non-coding DNA. Application will be species identification. Summary will be highly variable regions of non-coding DNA between rRNA genes are sequenced for species identification. And the last method that we can go for is whole genome shotgun method, where the target will be the entire genome and the application will be genome assembly. Summary will be entire genomic DNA is broken apart, sequenced and aligned based on overlapping regions to recreate the full original genome. Okay, so traditionally molecular methods such as sequencing of a microbe have been performed on culturable isolates grown in a laboratory setting. Sequencing allows detection of targeted genes as well as entire genomes of specific microorganisms. However, most microbes grow as a mixed microbial community, and potential biases become possible from the study of microbial genetic diversity, gene expression, and identification from a single species environment. These genomic methods have been used in forensic science to create genetic profiles for a single species and also for the identification of entire microbial communities. Genetic profiling can provide ancestral information on the microbial communities involved that can suggest if the strains are found in a certain geographical location or a laboratory, or provide profiles used to compare DNA found at a crime scene to a suspect or a victim. Narrowing down the locations is useful when investigating a criminal case so that time and energy can be concentrated on the correct tasks. Okay, moving forward. Sequencing of the 16S rRNA gene was one of the first targets for identifying taxa or organisms in the domains of bacteria and archaea and determining their phylogenetic relationships. The 16S rRNA gene codes for a single-stranded RNA that is a component of the 30S small subunit of prokaryotic ribosomes. Eukaryotic homolog can be used for identification. That can be used for identification is the 18S rRNA that codes for the 40S small subunit of the eukaryotic ribosomes. Both the 16S and 18S rRNA genes play an important role in the formation of the 70S ribosome and the 80S ribosome respectively, which are subsequently used in translation of protein synthesis. Okay, so let me just highlight few things over here for you. So, okay, let me first highlight and then I will explain. So, we all know that what are prokaryotes and eukaryotes, okay, and and there are different distinguishing features or different things which are present in prokaryote and absent in eukaryote, or there are many things that are present in both, but they are of different structures or different configurations. So, ribosome is one such thing. So, what happens is now, what is basically ribosome? Ribosome is basically the protein synthesizing machinery. Okay. So these are the things that I would like to highlight on this particular slide. So what are ribosomes? Ribosomes are basically the protein synthesizing machinery both for prokaryotes as well as for eukaryotes. So for protein synthesis, and we all know that the process of protein synthesis is known as translation, where from RNA a protein is synthesized. We all have that idea about the central dogma, right? That from DNA to DNA, that is replication, from DNA to RNA, that is transcription, and finally from RNA to protein, that is translation. So this is the basic, we can say a basic lifeline when we are talking about any living organism, whether it's a microorganism or a human being or a higher animal, lower group of animal, a plant, anything it is, any living organism, this is the central dogma that is followed. So now, depending on whether it's a prokaryote or eukaryote, over here we are talking about prokaryotes only because we are talking about microorganisms, and microorganisms are prokaryotes only. Okay. So as I mentioned, there are some definite distinguishing features between prokaryotes and eukaryotes. But if we talk about ribosomes, it's present in both prokaryotes and eukaryotes. But what is the difference? The difference is in the size. And we all know that ribosomes are having two subunits, one smaller subunit, one larger subunit. So together they are forming the, the ribosome. So in case of, in case of eukaryotes, we are having 40th subunit, which is the smaller subunit, and in case of prokaryotes we are having the 30th subunit, which is again the smaller subunit. So I will highlight this also for you. So for prokaryotes the smaller subunit is 30th, and for eukaryotes the smaller subunit is 40th. So together they are making what? 70th and 80th. This 16th and 18th. Okay, so 70th will be for the prokaryotes and 80th will be for the eukaryotes. Now when we are talking about this smaller subunits, there are divisions. So if we talk about the smaller subunit of the prokaryotes, which is the 30th subunit, it is having this 16S rRNA. rRNA means the ribosomal RNA. So what this 16S rRNA gene will do, it will code for this single stranded RNA, which will be a component of this 30th subunit of prokaryotes. While in eukaryotes, that same thing will be 18S. It will be doing the same job. It will be coding for the single-stranded RNA, which will be a component of the 40S small subunit of the eukaryotes. So for eukaryotes, it's 40S. For prokaryotes, it's 30S. For prokaryotes, it's 16S rRNA. For eukaryotes, it's 18S rRNA. And finally, if we talk about the size, then for prokaryotes it's 70s and for eukaryotes it's 80s. Okay. Moving forward, genomic method in forensic science. In forensic science, this method can be useful for quickly determining microorganisms associated with death from an infection using the presence of microorganisms to determine if the individual have been to a location specific for those strains, or to investigate if a person was intentionally infected. Say for example, COVID was for the first time detected as a epidemic in China. We all know that, and uh, still now there's a confusion. There is a group of people, a group of scientists who say that it was an accidental lab leakage from one of the labs. One of the scientists were working in a lab in Wuhan, China, and from there accidentally he or she somehow leaked that virus, and then it gradually spread. But there is again another group of people or group of scientists who believe that it was kind of intentionally spread like a bioterror agent, or bioterrorism, or biowar. So whatever it is, so since it was firstly detected in China, say for example during that period, I think for the first time it was detected in December 2019 in China. That's why the name was given as COVID-19. So if during that time, during the month of December of 2019, some people, so for example, a person had gone to China for whatever purpose, okay, as a tourist or for some work or whatever it is, and it was found that he got infected by COVID. So that is actually a location specific infection, isn't it? Because during that time only it was happening in China only, not in other parts of the world, not in India or Pakistan, right? So that is a location specific detection. So the intentional use of a pathogen to cause harm to an individual is called a biocrime. Again, I will just highlight it for you. So it's basically the same thing. Bioterrorism, bioterror, biological war, whatever. Ghuma fira ke, it's the same thing. While all microorganisms of a given species may have closely related DNA sequences up to 99.9% similarity, they are not exactly the same. This can arise from a phenomena called single nucleotide polymorphisms, or SNP, where a nucleotide has mutated into a different nucleotide base. The mutation may not affect the overall protein created, but it can be used as a marker for specific strains in a species or help determine the ancestry. The ability to determine the exact strain of a species is known as strain typing and is extremely useful in biocrimes cases. The identification of a microorganism is often a crucial component of an investigation, and the ability to identify microbes has improved significantly. However, there are multiple reasons why an organism can be misidentified even after sequencing. What are the reasons? Number one, microbial communities exchange DNA with each other constantly and have the potential to exchange a genetic forensic marker. Second reason, pathogenic strains and non-pathogenic strains can have different sequences. Third reason, databases can provide incorrect or incomplete organism identification as database accuracy has been shown to vary. So these are basically the different advantages and disadvantages when we talk about classical microbiological methods as well as molecular microbiological methods. So firstly, let's focus on the left hand side, that is classical microbiology. So what are the advantages? It's inexpensive, often specific, distinguishes between viable and non-viable organisms, can determine phenotype. Disadvantages: low sensitivity, training required, requires dedicated laboratory, often slow results, often requires pure culture. Now let's focus on the right hand side where we are talking about the advantages and disadvantages of molecular microbiology. So first let's see the advantages. It's sensitive, specific, distinguishes between strains depending upon the target, and it's also rapid. Disadvantages: it's expensive, requires strict quality control, requires specialized laboratory, requires high trained personnel, cannot distinguish between viable and non-viable organisms, requires isolation of DNA or RNA, requires personnel skilled in bioinformatics for the analysis part. Now talking about something called estimating PMI, that is the post-mortem interval, that is the full form of PMI. Current forensic analysis of body decomposition for estimating the PMI, the time elapsed since the death occurred. So basically post-mortem interval means how much time it has been since the death of that particular organism. These are mainly derived from taphonomy or in some cases using organisms, for example insects that have colonized a set of human remains. Time of death is an extremely critical part of the evidence that can include or exclude witnesses and suspects. The stages of decomposition follow an order of events that can occur over decomposition time. Stages of decomposition can be described as fresh, discolorization, bloat, active decay, and skeletonization. Okay, now talking about the stages of decomposition. Each stage is marked by certain characteristics such as mottled skin, livor mortis, rigor mortis. So livor mortis and rigor mortis, two are different things. In the fresh and discolorization stage, so livor mortis is basically when there is discolorization due to stopping of blood flow, and rigor mortis we all know when the relaxing and contracting power of the muscles is gone, so the muscle becomes stiff due to ATP loss, that is rigor mortis. Distension, that is bloating, purging of body fluids, and exposure and drying of the skeleton, which is known as skeletonization, that can be useful tools for narrowing the PMI. However, multiple factors like temperature, humidity, oxygen availability, sunlight exposure, burial, cause and manner of death, body composition affect how quickly decomposition occurs. Environmental factors like temperature and moisture have the most significant impacts on the speed of the decomposition process. So these are basically the different images that I have given over here. So A image shows 3 to 4 hours after death, showing signs of discolorization and livor mortis. B is showing 5 to 7 hours after death, showing rigor mortis, stiffening of the muscle as I mentioned. C is showing the remaining stages including bloating, active decay, purging of body fluids. E is showing drying of tissues later in the stage, and lastly F is showing natural mummification. Okay, now talking about microbial succession. So firstly, we will be talking about soil microbial communities. Cadaver decomposition results in the release of organic and ammonia-rich nutrients, altering both the pH and nutrient content of the soil, stimulating microbial activity. The area in which a cadaver decomposes creates a radius of nutrient and metabolic byproducts relative to the size of the organism, called the cadaver decomposition island or the CDI, that initially inhibits the growth of vegetation surrounding the carcass, means the dead body. The cadaver also attracts animal scavengers and insects that defecate or die in the area, continuing to add to the nutrient influx, which increases the microbial activity. Now, if you talk about mammalian microbial communities, mammals contain a large community of microorganisms that live inside and outside of their body. These organisms outnumber the host cells by tenfold and can provide benefits, opportunistic harm, or have no effect at all on the host. The microorganisms usually live in a specific niche on the host, for instance, bacteria that prefer to live in the nasal passages or others that prefer the colon. In the healthy individual, organisms remain in their preferred niche, but if a traumatic event happens, they can change the host's environment and immune system that allows the organisms to transmigrate to other parts of the body. Now, if you talk about studies involving human microbial communities, the use of human samples in microbial forensic investigations is still in the beginning stages of research development. Soil and animal surrogate studies have shown the potential for use of microorganisms as indicators of time since death or placement into an environment occurred. Firmicutes were the most prevalent among all body sites, though organisms from the phylum Proteobacteria and also bacteroid species were present. Samples from the lower GI tract and body cavity shared Clostridium, Lactobacillus, Aegirthella, and bacteroids, while Streptococcus, Provetella, Veillonella were found in the upper GI tract. The future direction for investigating the utility of microbes for measuring PMI will involve sampling from victims in current and future forensic cases with various manners of death and locations of death or discovery. Since current methods of estimating PMI will be used in these forensic investigations, it is imperative to estimate the PMI from the same cadavers using microbial succession so that the data can be compared for accuracy and reliability. The resulting data will also be validated by experiments conducted at decomposition facilities across the world to gain a database of decomposition in multiple environments and the changes of microbial activity associated with them. Microbial estimations of PMI along with current methods will allow investigators to construct an investigating timeline to make a stronger case against assailants or release potential false witnesses. So I think that's it. We will be soon moving on to the next second part of this particular topic in the next session. Till then, thank you and goodbye. Please like, share and subscribe iTailors Academy.